Welcome everyone. My name is Yiwei Li, currently a PhD student at the University of Western Ontario. Today I'm presenting our recent research, DELPHI, an accurate deep ensemble model for protein interaction sites prediction. This is a collaborative work between two Canadian universities. The co-authors are Brian Golding from McMaster University and Professor Yiwei Li from University of Western Ontario. So let's define the task of DELPHI here. DELPHI is a program that predicts protein-protein interaction binding sites. The input of DELPHI is the primary structure of a protein, which is basically the sequence. The output of DELPHI is the binding sites of that protein. Showing this in the screenshot at the bottom of the slide, the first two lines are the input protein name and the sequence in FASTA format. The third line is the prediction results of DELPHI on each amino acid, where 1 means binding sites and 0 means non-binding sites. We have tested DELPHI on 5 datasets. The training and validation datasets are constructed based on the testing dataset, such that there's no similarity between training and validation, and between testing and the union of training and validation. As shown in this slide, we use 12 features in DELPHI to represent a protein sequence. The first column is the feature names. The second column indicates whether the feature is computed using other programs or loaded as values directly. The third column is the dimension of each feature. Among the 12 used features, the first 3 are new, and the rest have been used in previous publications. The first new feature we use here is high-scoring segment pair, or HSP. An HSP is a pair of similar subsequences between two proteins. DELPHI will add a score to testing subsequence if it's similar with a subsequence in the training data, which also contains binding site. The second new feature we use is ProtVec1D. We developed this feature based on ProtVec, which was published in 2015. ProtVec uses Word2Vec to construct a 100-dimensional vector for each 3-mer. We collapse the dimension by making the summation of the 100-dimension vector, and we have experimentally verified this renders very similar results as the original embedding for this problem. And this way it uses much less memory comparing to the original embedding. The third new feature is position information. This position feature is inspired by networks like BERT, where the first position gives the value 1, second position gives the value 2, and so on and so forth. All features are normalized to a value between 0 to 1 before feeding to DELPHI. Now this slide shows the architecture of the DELPHI model. Each input sequence is firstly represented using the 12 features we just introduced, and then passed to the DELPHI model for inference. DELPHI has an ensemble architecture with 2 components, one CNN and one RNN. The intuition is that different components can learn different things from the input features, and combined together, they do a better job. The 2 components are trained separately, as shown on the left and the middle picture, and we use a fine-tuning process. The CNN model on the left has a convolutional layer, a max pooling layer, and fully connected layers with dropout. The RNN component in the middle has a bidirectional GRU layer and fully connected layers with also dropout. After tuning the CNN and the RNN model, we freeze the weights of the core layers, which are the convolutional layer and the bidirectional GRU layer. And then we move on to the right picture, which is the final ensemble model. We load and freeze the weights of the convolutional layer and the bidirectional GRU layer that we have tuned previously, and then we train only the fully connected layer. And that's the process of how we obtained the DELPHI model. The first performance comparison here is on dataset 448 and 355. We comparatively evaluated DELPHI with other 9 state-of-the-art programs. We have computed 8 different evaluation metrics: sensitivity, specificity, precision, accuracy, F1 score, MCC, area under ROC curve, and area under PR curve. Since binding site prediction is an imbalanced problem, which means the number of non-binding sites is much more than the number of binding sites. So MCC and area under PR curve will be a better indication of the overall performance. All numbers here are shown using heat maps. The darker the color, the better the performance. As we can see here, DELPHI has the highest performance in all metrics. It surpassed the second best program in MCC and AUPR by large margin. To further compare DELPHI with other programs, we used another 3 older datasets. And as you can see here, again, DELPHI achieved the best performance in all metrics. We did another experiment to show that the ensemble architecture and the 3 novel features improved the performance. We evaluated the CNN, RN, and the ensemble models separately, with and without the 3 newly added features. So in total, 6 tests. We plotted the value of area under PR curve and MCC of each test. And clearly, the ensemble model outperforms the individual CN and RN model. Also, the improvement due to the 3 new features is even higher. We have run DELPHI on the entire human proteome, and the result is available on DELPHI's website. DELPHI is open sourced. It's also available to use as a web server. Thank you very much for your attention.